Now we're going to have a closer look at the axon. After the axon emerges from the axon hillock, it can branch numerous times. Where the tip of a branch approaches a target neuron, it swells slightly to form a structure called a synapse. A signal is generated in the axon hillock that's propagated down all the axon branches to all of the target neurons. And this signal is called an action potential. It's a spike of electrical voltage, about 1/10 of a volt or 100 millivolts. But although it's a voltage, the action potential is actually an electrochemical signal that travels much more slowly than simple electrical conduction, only about 10 meters per second or so. An electrical signal, on the other hand, travels along a wire at an appreciable fraction of the speed of light. The speed of transmission along an axon can be increased by a myelin sheath. This myelin sheath is in segments, and the action potential propagates across the length of a segment by simple conduction, which is much faster. And the regular action potential is regenerated at the end of the segment and carried electrochemically across the gap to the start of the next segment. With a large diameter axon and a thick myelin sheath, the maximum speed can reach about 120 meters per second. Now, although this is still a lot slower than pure electrical conduction, it has the big advantage that exactly the same signal reaches all the target neurons down all the branches, even if there are thousands of branches. With regular conduction, electrical resistance would greatly reduce the signal that reached the endpoints. When an action potential arrives at a synapse, it triggers the release of a chemical called a neurotransmitter. This neurotransmitter diffuses across a narrow gap to interact with chemicals that are embedded in the membrane of the target neuron. These membrane chemicals are called receptors. The interaction between the neurotransmitter and the receptor affects the target neuron in various ways. One source neuron releases the same neurotransmitter at all its synapses, and different types of neurons release different neurotransmitters. However, depending on the receptors in the target neuron, the same neurotransmitter can have different effects. And to a degree, different neurotransmitters can sometimes have the same effects depending on the receptors. Three important neurotransmitters in the cortex are glutamate, GABA, and dopamine. Pyramidal neurons are called glutamatergic because they release glutamate at all of their synapses. And very often, the effect of glutamate is to increase the probability that the target neuron will produce its own action potential. And pyramidal neurons are therefore called excitatory. Other cortical neurons, called interneurons, release GABA at all their synapses. And very often, the effect of GABA is to reduce the probability that the target neuron will produce its own output. And interneurons are therefore called inhibitory. There are no neurons in the cortex that produce dopamine, but dopaminergic neurons in other anatomical structures extensively target cortical neurons. Dopamine can regulate changes to the strengths of glutamatergic synapses, or the degree to which a synapse will affect its neuron in response to one incoming action potential. Now, suppose that the axon from some neuron enters some target region. And in that region, there are dendrites of neurons that accept inputs from that source neuron. And I've illustrated one such dendrite. And the source neuron makes one synapse on one branch of that dendrite. But the same dendrite also gets synapses from lots of other source neurons. So that dendrite has to integrate information from all its sources to help determine if its neuron will produce an output. So in the next section, we'll discuss how all this information is transmitted and integrated.